anytime there. So now I have the pleasure of introducing Vivian. Dr. Vivian is a researcher that has taken a from JdVS, and he's going to share about his research and the mouse models that he created and are growing. Very fascinating, very interesting, so we're very happy to have you here.
Okay. Well, thank you everybody for your participation, and thank you very much for the invitation. I'm very excited to, it's my first Janssen-DeVries Syndrome Foundation meeting, so excited to share our work, which is unpublished. And so just want to give you a little bit of background. So obviously, my laboratory has interest in rare diseases, and I'm at Children's Mercy Hospital in Kansas City, and there's many of you are very familiar with this idea of just the need to study rare diseases. And in particular, our particular interest is because many of these diseases, and Janssen-DeVries syndrome is one of them, that they're poorly understood. And importantly, there are no treatments. And so the reason why we study rare disease, particularly at our pediatric hospitals, are rare diseases disproportionately impacts children. So I got involved with this through some early discussions with a local family who eventually did get diagnosed with Janssen-DeVries syndrome, and that's how we initiated this project. So my background is I'm a mouse geneticist and a pluripotent stem cell biologist. So my laboratory is a bench laboratory program where we use the mouse as a model system and human-derived pluripotent stem cells as systems to study and understand rare diseases. Why do we use those tools? Well, it's a chance for us to understand the diseases better in a research laboratory setting, and also for understanding the potential for identifying new therapies. And so we go back and forth between our animal models, which we're very cautious about using and use them only when we feel as if they're needed and important out of consideration for animal welfare issues, and moving back and forth between the animal models and the human pluripotent stem cell models. We feel as if they both can provide some tremendous interest or powers and interest to study rare disease and to identify new therapies. And what we do is we take advantage of our genetic engineering technology. So ever since I was in graduate school, I've had interest in making genetic modifications in the mouse, and I continue to do that. And so we have some advanced technologies where we can genetically engineer mice that carry the precise pathogenic variants that are present in a particular disorder. And we have that capacity. We study that obviously in the whole animal. Whereas human pluripotent stem cells, which can be human-derived, and I'm not going to talk about that today, allow us to leverage a human cell line derived model for growing in a dish in a laboratory. Now for Janssen-DeVries syndrome, we focused on the animal models. We haven't looked at all into any of our human-induced pluripotent stem cells. Dr. Lockman, who you guys will hopefully go into the YouTube page and follow his presentation, has some very interesting research involving human iPS cell models of JdVS, but our focus is on using the animal models. And so Dr. DeVries gave a background on the nature of the mutations that are present in Janssen-DeVries syndrome, and they're in this gene called PPM1D. What I've done is I just diagrammed if you stretch out the protein, or the thing that is encoded by the PPM1D gene, and sort of stretch it out, you can see where the mutations tend to arise. And these little red stop signs that I put are actually the mutations that were initially identified in Dr. DeVries' initial paper from 2017. And what that ends up doing, and Dr. DeVries gave a nice background on this, it ends up truncating the portion of the PPM1D protein. So in other words, part of the protein is made, but part of it is missing. And so these are dominant gain-of-function truncating mutations. And so PPM1D is not a new gene. It had been studied for quite some time, and we'll talk about in other contexts. And there had been a knockout or a null mouse mutation, or a mouse that doesn't have any PPM1D in it. But we were very interested in identifying or generating an animal model that actually recapitulated these types of variants that are seen in Janssen-DeVries syndrome, which had not been made at that time. And so those were kind of our early goals. The other reason why we had some very early interest on this is that there had been already information that there were somatic mutations, in other words, mutations associated with cancer in this same gene, and they tend to cluster in kind of the same region. And they're not very common, but they are present. And so if you actually, and this is actually a diagram sort of overlaying where those sort of hot spots are that are seen in mutations in certain, in some cancers in the PPM1D gene. And they don't exactly overlap, but they're in the kind of the same region as those truncating mutations that Dr. DeVries had first identified. So we had some, interest that, would there be any propensity for tumor, for cancer in these children with Janssen-DeVries syndrome? And we wanted to ask that question in our animal model. And I'll give you the short answer is there is no evidence of this in our animal model. So I think you can rest assured it's a negative result, but we don't believe that there is an increased propensity for cancer. But more studies need to be known. So what was the genesis for this idea or this concern? Well, PPM1D, it had been known what its function is as a phosphatase. So Dr. DeVries mentioned this. So a phosphatase is an enzyme that's in our cells that can erase the phosphates groups that can be tagged onto other proteins. And what's interesting is PPM1D is known to erase the phosphates on quite a few genes that are involved in cell cycle arrest, DNA damage checkpoints, and these are genes associated with cancer. And particularly the central protein I'm showing you here is P53, which is generally thought of to be the guardian of the genome. And so this is why there were several labs who had studied PPM1D previously, but more in the context of tumor development. The idea that PPM1D can actually turn off these genes that keep cancer off. And so by activating PPM1D, potentially it could cause some propensity for tumor development. And so those were some of our concerns early on that we wanted to model in the animal model. And so our objectives were to develop some mouse models for Janssen-DeVries syndrome, and then in particular, identify what their propensity is for tumor development. And the idea is that we could both understand the disease, address that question of tumor development, but also use it for some other studies to demonstrate how good of an animal model is representing the various aspects of Janssen-DeVries syndrome, with the future idea that we could possibly use them to both understand the disorder more, but possibly to identify and develop new therapies. So the question came about is we have capabilities of genetically engineering a mouse just about any way we want to. And so the first question was, well, what mutation do we want to make in the mouse? And so I worked closely with my collaborator, Dr. Tomoyo Okuma, who is our cancer specialist, and he had a desire to make the mutation, of course, in the cluster that's more preponderant in the tumor, whereas I wanted to make one that was much more preponderant in the cluster associated with Janssen-DeVries syndrome. And we couldn't agree, so we made both models. So we made two mutant mouse strains, one that has a truncation right in the middle of that cluster that Dr. DeVries had first identified, and then one, which was W427X, so it's a truncation after the tryptophan, and then a second one which truncates itself at amino acid 512. So we have two different mutant mouse strains, and we've done a little bit of analysis, but more of the focus is on the Janssen-DeVries mouse model. So to ask that first question is, well, are these mice tumor prone? We just let them live for an extended period of time. This data takes us out to 80 weeks of age, which is a fairly old mouse, about a year and a half. And we saw no evidence that they developed any tumors at all in our Janssen-DeVries mutant mouse model. And so that was very reassuring. Additionally, what we did is we combined the mice that carry the Janssen-DeVries mutation, PPM1D, along with a tumor-prone model, that P53 gene that PPM1D is known to interact with and activate. And that mouse model is tumor-prone. What we saw was there was no what we call an exacerbation. So in other words, the tumor development that occurs in that PPM1D mutation is the same if we combine it with, let me back up. The P53 mutation develops tumors. That doesn't get more severe when we combine it with the Janssen-DeVries mutation. So that was a negative result, but that was very reassuring that we saw no evidence of enhanced tumor development in this animal model. And this is what we call a Kaplan-Meier plot, just looking to see when we had to remove the animals from the study because they were sick. So that was reassuring. So we don't have any evidence that these mice are tumor prone, but they had some interesting phenotypes that I think are very relevant to Janssen-DeVries that we're still continuing to study. The first off is that the mice are smaller. In our parlance, we call them runted. And so they're quite smaller. And of those two mutations, the W427X, the more Janssen-DeVries-like one, tends to be quite a bit smaller. And so this is just a growth curve compared to wild type in black. The red line shows the weight of of our mutant mice. And so they're small, and they're smaller throughout their entire lives. What's also interesting is they're actually smaller. When we combine that mutation with that P53 mutation, that actually makes them even smaller. So even though they aren't prone to any tumor propensity, there is a genetic interaction between PPM1D and P53, which is interesting. So PPM1D it's clearly doing some functions of P53 outside of its tumor development, but it does make the runting more severe. What we're really interested in, though, were the neurodevelopmental aspects of this. And so one of the great things we can do with animal models is mice do things. They walk. They hide in little holes. They like doing things. And we can monitor that, and we can measure it in the laboratory. And so I want to show you a couple of these. This is called behavioral phenotyping. So here, I want to show you a couple little tests we've done with these mice. The first one's a beam walk. So the first one is pretty straightforward. We put a mouse on a little beam, and he likes to run over to the far left-hand side of that box because he doesn't like being out in the open. And we can measure how well they can navigate that thin beam. It's only about a centimeter and a half in width. And so we can measure how quickly they walk and how many times they slip. So it's a measure of coordination and motor function. And so what you can see and appreciate here is our mutant mice take a lot more time to get across their beam. So there is clearly some deficits of motor function and coordination in these mutant mouse models. And that lasts, at least in the more severe mutant allele, that lasts throughout their entire lives. So that we are showing some deficits in motor function. Another assay we want to learn from is what is their capacity for memory and learning. And so this is a Barnes maze. So what this is just a flat table with a whole bunch of holes around the periphery. And underneath one of those holes is a little box. And the mice, when you put them in the middle of the table, they don't like being out in the open. If you ever have a mouse in your house, they don't stand in the middle of the kitchen, they go onto the edge. So they move around to the edge and they find that little hole and then they hop right into it. Well, if we keep that hole that's covered in that little black box in the same spot, the mouse learns where that hole is. So we do the same test day 1, day 2, day 3, day 4, day 5, and the mice can remember where that hole is. And they get better at finding that hole every day. And you can see in the wall-type mice in that white circle, they take less time to identify where that hole is. When our mutant mice, they're delayed in being able to identify that hole. So that's a measurement of memory and learning. And so relatively mild deficits, but I think this Barnes maze is really pointing to some mild deficits in learning and memory. Another assay I'm very interested in, although it's hard to interpret, is what we call a marble burying assay. So if we put a mouse, we take a whole bunch of black marbles and put them into a fresh mouse cage, and we put a mouse in there, they're fascinated by it. They love to dig around and dig into the bedding, and then after about 30 minutes or so, a whole bunch of those marbles become buried. And that's part of their burrowing behavior. So it's an easy assay because you leave them in there for 30 minutes and you just count the number of marbles that are not buried or not buried. And so it's thought to be, it's difficult to interpret what it actually means, but it's thought to be involved in measurements of certain compulsive behaviors. Mice just like doing it because they find it fun. And so it's just something that they want to do, and that's called a compulsive behavior. So it could be a measure of possibly anxiety or an obsessive compulsive behavior. And what was really striking is in comparison to their wild-type siblings, our Janssen de Vries mice do not bury their marbles. You can see on that first table, they just don't do it. And so for reasons that we are not really fully understanding, but is a strong phenotype. And it's not because the mice are just not moving. You can imagine if they're hiding in the corner and not doing anything. We put them under an actimeter, which is an open box which just measures general activity. They're actually more active than their wild-type siblings. So it's not like they're not moving around. They're just not burying their marbles. So it's a fascinating behavior that I think is something we can really follow up on. And so I've tried to sort of identify some of these behavioral phenotypes that I think have some links to what we know, but still need to further understand with our Janssen de Vries children and individuals. And so we have a lot of directions we can go towards. And so, but I think we've got a useful and very interesting model that we can further understand Janssen de Vries syndrome. And so I definitely want to do some more molecular studies to really try to understand what is the underlying molecular deficits associated with the central nervous system tissues in these animals. And also look at some proteomics, which is studying how the other proteins that are present in the tissues are affected by this this type of mutation. That could start identifying where we could start pointing to new directions and therapies. Dr. Lockman, I was hoping we'd be able to hear his presentation, but he also has some interest in a specialized cell type in the central nervous system called the microglia. And so he wants us to get some microglia from our mouse models. And so we're going to start, we're pretty good. We have interest in microglia as well for some other very severe neurodegenerative disorders. And so we're going to try to get some microglia over to him derived from our animal models to try to mimic or try to parallel what he's studying in his human iPS cell models. Definitely want to do some deeper behavioral analyses. We can do a lot of really neat things, particularly our colleagues at KU Medical Center for this. Another very interesting thing, which really got me interested very early on, is because PPM1D is, the protein is a phosphatase. So a phosphatase is an enzyme. Enzymes are druggable, and there's actually already several drugs that are known to modulate PPM1D's activity, in part because of the tremendous interest in this protein in tumor development. So we can kind of piggyback off of what people are doing in studying this gene and this protein for cancer to possibly consider what is its utility for possible therapeutics for Janssen-DeVries syndrome. And I've just highlighted a few of these drugs, compounds. I shouldn't call them drugs. These are not FDA-approved drugs, and so they'd be exploratory drugs. But I think there's some things we can study by combining the quantitative behavioral assays that we have and hopefully molecular assays that we have in our mouse models with applying to some of these therapies to see if they could be potentially useful. So very interested in that. And we also, I'm a carpenter, so everything looks like a nail with my hammer. So I want to make some other mutant mouse models where we can remove or alter PPM1D function in specific cell types. We've got a model for that needs further study. So I'll go ahead and stop there. I hope it kind of gives you just a little flavor of some of the things we'd like to do and how we can leverage the power of our genetic engineering tools to really understand the disease in a better way, particularly in a preclinical model such as the animal model. Definitely want to acknowledge my laboratory, my collaborators at KU Medical Center. I actually started this work when I was at KU Medical Center. I've since moved to Children's Mercy Hospital in Kansas City, but our collaborations continue. And I do want to recognize some of our funding sources that we've gotten to start this little pilot project. So really appreciate your time. A little different of a talk from what we've seen yet today, but happy to answer any questions. So thank you very much.
Can you go a little bit further into the purpose of having phosphatase and the phosphatase action around this to simplify a little bit?
Okay, yeah, so PPM1D is a phosphatase. So it's in most of our cells, and it just normally acts to erase the phosphates that are on other proteins. So phosphates are little, we call them post-translational modifications. They're little tags that get placed onto certain cells. And those phosphates are important because they can control that protein's activity. So by PPM1D being able to potentially erase those phosphates, it can, post-translationally, it can control the activity of that protein. So PPM1D is a regulator, if you will. And so its probably job is to balance out, for example, P53's activity, because they don't want too much P53 or too little P53. So it's kind of, it's one of these regulators that's helping control normal cellular homeostasis. Probably what's going on in Janssen-DeVries syndrome is that homeostasis in neurons and other cell types is disrupted because of that activated, that enhanced phosphatase activity that's associated with that mutation. We don't understand that well enough yet, and that's what our preclinical models are meant to say, or to address. Does that answer your question? Okay.
Jay, can you talk to us about any social activity that you've seen in these mice?
The short answer is that we haven't measured it yet. There are a wide range of behavioral testings that we haven't done yet. But social ability is one, particularly now that I've met a large number of kids with Janssen-DeVries syndrome, I want to do that. A very simple test that you do is you can take another mouse, put him in like a little cup that's got like little holes in it, and then put the mouse that you're testing, the Janssen DeVries mouse, and put it next to it. And you can actually measure, mice are curious, they want to interact with their friends, and there are models where mutant mice do not want to interact with their the little mouse that's in the cage next to them. I would love to do that test. So there are a variety of social interaction tests that we hadn't touched. We're focused more on just baseline, motor function and neurological, but there's a lot of good things. And these kind of come a little bit beyond my ability, but where our collaborators could certainly apply. But I would guess what the phenotype would be. We would let the mice tell us what their phenotype is.
Okay, two quick questions. The one where you were showing those marbles, and it was saying that that's kind of related to maybe obsessive compulsive. Is that if they bury the marbles or if they don't?
It's what you bring up is a really good question. Is the result is not exactly consistent with how I would interpret that marble burying test for obsessive compulsion. It is, so I cannot explain the actual results other than as a very dramatic phenotype and how that relates to whatever is going on in the brain of this PPM1D mutant mouse. They're just not wanting to do that rooting behavior, and I don't know why. So your question, thank you for catching that. The actual phenotype we see would actually be inconsistent with what I would predict based on what the clinical phenotypes are, but the data are what they are. It's measuring something. We're not sure what.
Very interesting. And then you had shown those a couple exploratory potential drugs.
Uh-huh.
Are those specifically exploratory drugs that are being made for JdVS to use in the mouse types or just in other conditions?
Those are drugs that are being looked at more in the cancer angle for turning off PPM1D in certain tumors. But it would be intriguing to apply those to our JdVS models. It's always valuable to piggyback off of other people's work, particularly cancer, which has a lot more money than some of our rare disease work.
Thank you.
Yeah. None of them are FDA approved, though, so I would hope that the cancer folks would continue to push hard for that, because there are a subset of tumors that are clearly activating mutations for PPM1D. Not many, but they're fairly rare.
When you were looking at the mice sizes, are they clinically significant, or is it just like a point of interest?
They're statistically significant. So the mice are smaller than their wild-type counterparts, and so it is statistically significant. In terms of clinical significance, there's no animal welfare issues associated with them being small. They're just kind of small, but they're statistically small. Does that answer your question?
And then, are there plans to, additional generations continue on that research and see what their offspring might look like? Or
We've looked at several generations, and they all have the same phenotype. It hasn't changed over time, which is reassuring. It shows us we've got a consistent genetic phenotype.
Are there any seizures or vomiting or tremors?
A good point. We haven't seen seizures, but it means we haven't looked at them that closely. The vomiting thing gets brought up a lot. Rodents do not vomit. That is a clear and strong phenotype. There's a side story I could tell you about how that is important. And so the question always is well, can we use this to monitor or understand cyclical vomiting? Even though rodents don't vomit, the underlying biology is still there. And so there are clearly very good, strong mouse models for migraine. And so I think some of the underlying biology we can understand, even if we don't get the ultimate output of vomiting in a rodent, which just doesn't happen. We haven't looked at it, but I would not expect us to be able to see vomiting in a rodent because they don't do it. Okay? All right. Thank you, everybody.
All right, guys, at this time, we're going to take a